we get experimental science. We're curious, non-judgmental.
Gwen did have suggestion that these were the same species when we last talked about them last week, but maybe having some imagery, more imagery associated would be a good way of following that up. So Rose, these ants look very different, right? You see the vertical mandibles, right? Those are extinct and they don't exist anymore. Those vertical mandibles have now turned into the horizontal ones. So that's the biggest difference in terms of the function and form of the animal and how it looks. We're about to look at the hell ant specimen under the microscope. This is 90 million years old, and it's gonna be a lot of fun. This is what the shape looks like. Extinct animal. We have two of the samples we're gonna look at underneath the microscope in just a moment. Think mama is just how they look. I think the first time someone found them, we're like, this looks really scary. I'm gonna call it the hell ant. So literally, Ian, that 20 million years, there is actually another 40 million year gap in the fossil record. So it could have been as long as 60, but there is this massive gap in the fossils where it's like they're around at 90 million years, and then the next batch of fossils that you find are 50 million years old, and you don't find anything in between. And so we don't know when in that gap they went extinct. We were talking about the content of the tree, the resin, the amber, and that's a kind of tree sap. So while of what may have been utilized for, as Cliff was saying, of nutrient transport, it also is used to seal injuries. So like if the tree is cut, it's a way to seal off like openings and wound sites for the trees, and that's something where the insects could get caught in. And what's cool with this image, y'all, is that this website that has that same specimen here, and Gwen, correct me if I'm wrong, I think this is the one that we think is the species that we're also looking at here, Hadromimac. There's some really cool and beautiful specimens. You can see different lengths of the mandibles as well that they have. We've got a pretty good mandible length on ours. Like that they have this plasticity where they can move between different castes. They could be a worker or a queen. There, in those Harbagnathus, you'll have regular ants that are the queen, or the workers that can become a queen. But then they occasionally have a true queen that's hatched from an egg that has wings, and the idea is it can fly off and disperse its genetic information. So it's not wild to think that these animals also have that. And then the next bet would be, okay, maybe then they also have the ability to transition between worker caste and then queen caste. But the only way to prove that in amber would be to have a hell ant without the wing nub caught laying eggs. And I don't know how, but we're lucky enough to have a sample of a fruit fly laying an egg in amber. You would need that for the hell ant, and you'd need the non-winged version doing it. And so these are just confusing, right? Because if you're trying to say that these animals live in colony settings, and then you only find amber with one hell ant and then other species of ants, it's like, whoa, what does that actually tell you? So it's the frustrating part about, researching something that's extinct, and you're trying to find evidence and information based off these snapshots. It doesn't necessarily mean that they live in colonies, though. Yep. Oh yeah. Ian, it could very well be the alternative hypothesis, right? Is that these are more solitary ants, just like solitary wasps, and that's maybe how they live, right? So like Ian is doing this right, y'all. Being curious and asking questions and be like, wait a minute, it doesn't quite make sense. Where's the evidence to suggest that this is social? I think, Ian, honestly, the only evidence is that it looks ant-like and it has some of these metapleural gland and some elements that we know are in social ants today. So there are no solitary ants, Rose, but they are ants and bees are descendant from wasps, and there are solitary wasps. So this could be a transition point, right, in between wasps and ants, where you're maybe like a little bit more ant-like, but in some respects you're still like a wasp. You just don't know based on the evidence that we find, right? See, this is that big gap that we have, Ian. And so we just don't know. There's a gap in where certain ants are coming out, and then there's a gap in like when we see the last hell ant. So in terms of how long they've been alive, because you were asking earlier, we don't know. There's a general gap of the ant fossil record overall, and then there's a bigger gap for hell ant fossil record as well, because there are just so few samples. Like Rose, if you look online, try googling hell ants, there's not a lot that are just like available floating around, even in terms of images for you to look at. So it makes the specimens really important to preserve and to analyze. Research is interesting. There are no other insect species that have vertical mandibles similar to hell ants. Yep. Isn't it cool, Ian? So that's why, Ian, too, I have a sneaking personal hypothesis that huge gap in the fossil record, that 20 million plus years, you need to have there must be transitional states, right, that goes from the vertical mandibles to horizontal. Like, it just doesn't happen overnight. But I have seen nothing that actually represents a nice transitional state. It's like different parts of the world were good at different times. And so then not only is there gaps in ant fossil record, but at a particular location, there's massive gaps as well. And so how do all those things connect? It's like, well, then how do you really know how things work and how they not? All right, chat, without further ado, we're going to fire up the microscope and we are going to look at our Helland specimens. We have 2 of them today that we're looking at, which I'm super excited about. Y'all take a look at this region here, all those little hairs coming down. That is similar to these little hairs coming down from the carpenter ant. Right, so that's that same structure, that's that tongue that the animal uses to eat. So on this one, and Andrea, let me zoom out for you first and give you a little bit of a comparison. So look at this sample here in its zoomed out state versus this one. So Andrea, one way that you can tell the difference between Dominican amber and the Myanmar amber is the color. And so you see how this looks like a drop of honey and it's very clean. And most of the samples are gonna be very, very clean like this, where it's gonna be, you know, see-through. Like, there might be, when we get discoloration in the amber in terms of, I mean, like this here, that's because the insects leaked some hemolymph or blood into the amber and it like messed up the resin colors, versus here, this is like what the natural resin would look like. If you look at the resin from the Myanmar region, this is what it looks like. You see how dirty it is? There's just like a lot of stuff in it. So this is one feature that you can use actually to identify what part of the world the resin came from. Clear as a honey drop, it's the newer amber from the Dominican Republic. If it is more like still yellow but almost schmutzy, then it's like the Myanmar region. And I think it's probably just what the ecosystem was at the time, what trees were the sap was coming from, etc. So we don't see the cells in this, but what you can see, Lou, that is a great question. Lou, this is 90 million years old. So we have no idea what the predator was, if any, of this ant. We know what it ate, which it did eat small cockroach-like creatures. And the only reason we know that is because we caught some specimens in amber of it. Is that a good, you know, that's like one piece of its diet that we know. We have no idea, though, what, if anything, actually ate this creature. So, Ian, that's what I'm not sure about. Is this actually serrated in a way, or is it damaged and the tips have frayed? Or are there like sensory bristles on there, right? Like, is there a biological reason for this looking like this? Or is it just a result of, yeah, like when it was drying, because it's a thin tissue, it had some cracks to it and frayed? I don't know, Ian. All right, let's take a look on the other side. So Ian, we can still see the spikies here, so we'll zoom on in. You can actually, Ian, do you see how cool this is? You can see the fur, little hairs on the leg. From this angle, it looks like sensory bristles, right? So if you compare to the bristles on the leg, it just looks like it's a lot of bristles on top of the head as well on that horn. All right, Ian, that's probably the best in terms of lighting that we'll have. Looks like the horn connects to the antennae. Yeah, there is this figure where they're looking at this old state. There is some internal tissue, Nin, but it's like a dried up internal tissue. So we don't actually get any like detailing from it. So if we look at this, like if you look at C, you know, that could be like one possibility that we have here, right? So I think there's a gap in between the horn and the antenna. And actually, one cool thing about this one, so I can do a thing called blowing out the image, which is making it super, super bright. But what you can actually make out is, look at this, you can actually see the eye now. The reason this doesn't show through otherwise is because it's just so dark that the light doesn't shine through. But when we're turning up the exposure, you can actually make out where the eye is. And then here, right, there's some of that dry cuticle within. So this is the second sample, is this one. So what's weird about this one, Ian, is can you see how much more clear it is? It's missing its innards, as it were, so it's much more of the exoskeleton versus the rest of the organism. And that's if we down just a little bit. See, look at that. So that is the abdomen split. But it does give us a better glean into more of the detail of the animal. This one, Ian, is super cool because here you can see the eye cells. So there are the hairs, again, on the horn. Seems, again, like that. Either sensory bristles or fibrous, or I don't know. There's something definitely going on there. Our suspicion, Ian, is that the last time we looked at these two, that they were the same species. The two different samples that we had. See, the mandibles here, so it's almost like if we didn't know to look for them, you could miss them, but there they are, looking much more empty than before, right? So if we compare to this ant, right, there's much more internal anatomy, like it's dried away, but you still see it. Versus here, this one like almost looks empty. Let's go ahead and turn it over. The size of the eye is more indicative of their visual animals, like in terms of what they hunt or if they're underground dwelling. But yeah, so there you can see the eye cells, all those little circles, right, are the ommatidia. Ommatidium, singular, their neurons go into the brain of the animals. Yep, have the brain like processing information. It's a really nice image of it on this side. And yeah, you can see there's maybe some tissue that's kind of fallen deep into the animal, but it's mostly just exoskeleton here. That's one of the reasons I like this specimen in contrast to the other one that we have. We're actually getting two nice different views of the animal.